Welcome to my YouTube channel. In this video, we are going to learn about hemostasis. The hemostasis is the process at which the blood clot is formed at the site of vascular injury. Normal hemostasis process involves platelet, clotting factor and endothelium. They all, that is platelet, clotting factor and endothelium, culminate in the formation of a blood clot which prevent or limit the extent of bleeding. The sequence of events that occur in the process of hemostasis are arterial constriction, primary hemostasis, secondary hemostasis, clot stabilization and resorption. In arterial vasoconstriction, the blood flow is reduced to the injured area. The endothelin released from the endothelial cells, that is endothelin released from the endothelial cell, is a vasoconstrictor which helps in the constriction of the artery. This is actually a neurogenic mechanism and is a transient effect. In primary hemostasis, platelet plug is formed when the vascular injury occurs. The endothelium is disrupted. Then the exposure of subendothelial von Willebrand factor and collagen will promote the platelet to get aggregated. When the platelet altogether aggregates, then the platelet plug is formed, which is called primary hemostasis. Next is secondary hemostasis. In secondary hemostasis, there is deposition of fibrin. When the tissue factor binds with factor 7, thrombin is generated. Then the thrombin cleaves the fibrinogen and forms fibrin. The fibrin altogether get polymerized and form a meshwork, which is secondary hemostasis. Next is clot stabilization and resorption. In clot stabilization, the fibrin formed during the secondary hemostasis binds with the platelet and form a solid permanent plug. Then after some time, the clot is resorbed by the tissue plasminogen activator produced by the endothelial cells. We have already seen that the process of hemostasis involves the platelet, coagulation factors and endothelium. Now first we are going to see about platelet. The platelet helps in the formation of primary plug, that is we have already seen in the primary hemostasis. It helps in the formation of primary plug and helps in sealing the vascular defect, and the platelet provide a surface that bind and concentrate the activated coagulation factors. The platelets are nothing but disc-shaped anucleate cell fragments of megakaryocytes in the bone marrow. There are two types of cytoplasmic granules, alpha granules and delta granules. The delta granules means dense granules. The alpha granules have P-selectin, factor 1, 5, von Willebrand factor, platelet derived growth factor, fibronectin. And the dense granules, that is the delta granules, have ADP, ATP, ionized calcium, serotonin and epinephrine. Primary hemostasis, that is platelet plug formation, have three steps: platelet adhesion, platelet activation and platelet aggregation. In platelet adhesion, by the interaction between the platelet surface receptor glycoprotein 1b and von Willebrand factor present in the subendothelial matrix, the platelet adhesion occurs. The platelet is also adherent to the collagen present in the subendothelial matrix via glycoprotein 2a. Then during platelet activation, the shape of the platelet is rapidly changed. It changes from a round disc to a flat plate with spiky protrusions so that the platelet surface area is increased. So when platelet surface area is increased, more other platelets can come and aggregate to each other. Here the secretion of the granules, we have already seen about the alpha granules and delta granules. So the secretion of the granule contents occur together with the change in shape of the platelet. So the change in shape of the platelet and the secretion of the granule contents, both the process together called platelet activation. The thrombin activates the platelet and in turn the activated platelet produce prostaglandin thromboxane A2, which is an inducer of platelet aggregation. So the next process is platelet aggregation. In platelet aggregation, the platelet have a receptor called glycoprotein 2b and 3a, to which the fibrinogen is bound. So the fibrinogen bound to a platelet bridges with the adjacent platelet leading to aggregation. In this picture you can clearly see that the glycoprotein 2b3a receptor of the platelet is bound with the fibrinogen, which is bridged to the glycoprotein 2b3a of the adjacent platelet. So the platelet all together gets aggregated. This all finally leads to the formation of platelet plug. This is called primary hemostasis. Remember a point here, when the glycoprotein 2b3a receptor of the platelet is deficient, it results in the bleeding disorder called Glanzmann thrombasthenia. The initial wave of aggregation of the platelet is actually reversible, but later the thrombin will stabilize the platelet plug and it will become irreversible. Then the secondary hemostatic plug is formed. Next is about the coagulation cascade. Series of enzymatic reaction which leads to the deposition of or insoluble fibrin clot. Each step of the coagulation cascade involves an enzyme, a substrate and a cofactor. An enzyme is actually an activated coagulation factor and substrate is a proenzyme form of coagulation factor and a cofactor is actually an accelerator. A coagulation cascade usually have two pathways, extrinsic pathway and intrinsic pathway. The extrinsic pathway is based on prothrombin time and intrinsic pathway is the partial prothrombin time. The extrinsic factor is depending on the blood vessel injury, the factors released by the vessel wall, and the extrinsic factor involves the factors released by the tissues. Thrombin have most important activities. Some of them are given here. Conversion of fibrinogen. The conversion of fibrinogen into a cross-linked fibrin is an important function of thrombin. Next is platelet activation. The thrombin is a potent inducer of the platelet activation and aggregation. Next is pro-inflammatory effect. The thrombin have a pro-inflammatory effect on inflammatory cells, endothelium and other cell types which contribute to the tissue repair and angiogenesis. Next is the anticoagulant effect. When the thrombin encounter a normal endothelium, it changes from a procoagulant into an anticoagulant. That is, beyond the site of injury, the thrombin is converted into an anticoagulant. That is the anticoagulant effect of thrombin. Next is about the endothelium. The endothelium usually have prothrombotic activity and antithrombotic activity. Here we are going to see about the antithrombotic activity. First is the platelet inhibitory effect. The factors which are involved in platelet inhibitory effect are prostacyclin, nitric oxide, which is synthesized by the endothelial nitric oxide synthase enzyme, and next is the adenosine diphosphatase. This enzyme helps in the degradation of the ADP. And next, anticoagulant effect. The factors involved in the anticoagulant effects are thrombomodulin, endothelial protein C receptor, heparin-like molecules, and also tissue factor pathway inhibitor. Next is the fibrinolytic activity. The key component of fibrinolytic pathway is tissue plasminogen activator, which is synthesized by the endothelial cell. Like, share and comment my video. Do not forget to subscribe my YouTube channel. Thank you for watching.